Assalamualaikum warahmatullahi wabarakatuh. Good evening, everybody. Can you hear me?
Yes.
Yes, clearly.
Yes, okay, thank you. Alhamdulillahirabbil alamin wassalatu wassalamu ala mussafil anbiya'i wal mursalin wa ala alihi ajma'in. Good evening, everybody. Honorable Associate Professor Dr. Malgorzata Korzeniowska. Sorry if I not clear, write your name. The speaker of general lecture, Honorable Professor Dr. Insinyur Elisa Julianti, MS, Head of Department of Food and Science and Technology, Faculty of Agriculture, Universitas Sumatera Utara, Honorable Dr. Insinyur Tapi Supriyana, MSI, Dean of Faculty of Agriculture, Universitas Sumatera Utara, and also all of us guests and our loved students. Ladies and gentlemen, may I first take this occasion to pay tribute to all of participants of this general lecture of Department of Food Science and Technology, Faculty of Agriculture, Universitas Sumatera Utara. It is a great pleasure and an honor for me to deliver this welcome remark at this opening ceremony of general lecture of functional food, with the topic is bioactive compounds from meat and meat products. May I first take this opportunity to express my gratitude and appreciation, as well as extend a cordial welcome to all of participants for attending this important event, and especially to our speaker today, Associate Professor Dr. Malgorzata from Department of Functional Food Products Development, Faculty of Biotechnology and Food Science, Wroclaw, University of Environmental and Life Science, Polandia. I wish you a hug, welcome, and a presence join in this Zoom meeting. Ladies and gentlemen, and all of our students, this general lecture was designed to provide opportunity of shared knowledge and experiences, exchange information, and research in functional food of bioactive compound from a meat and meat product. I do hope that this general lecture able to give us a new insight of view of the topic. So please, take a big benefit from the general lecture. You can make a new networking and maybe sharing knowledge, and all of you can get from this lecture. To conclude, at last but not least, I would like to extend my appreciation to the organizing committee from Department of Food Science and Technology, Faculty Agriculture, Universitas Sumatera Utara for making this event a success. And finally, I wish all of you to enjoy this program, enjoy your meeting in this virtual meeting, your lecture in this evening. And let's pray that all our event remain in this blessing of God Almighty. Amin. I think that's all. Wabillahi taufiq wal hidayah. Assalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh. Thank you, Dr. Risawita.
Thank you so much for Ms. Dr. Insinyur Risnawita SP MSI for the stunning opening remarks. Ladies and gentlemen, please welcome our moderator today, Mr. Jonser Naibaho, STP MSI, alumni from ITP USU, batch 28, PhD candidate in Wroclaw University of Environmental and Life Science, Polandia. For Mr. Jonser Naibaho.
Okay. Thank you very much. Can you hear me well? I hope.
Yes.
Before we start, Prof. Gosha, maybe later I can speak in bahasa a little bit. So later on, nanti kalau kita ngikutin kuliah, nanti kalau misalnya ada pertanyaan yang kalau bisa pakai bahasa Inggris boleh, tapi kalau misalnya pakai bahasa Indonesia juga boleh. Terus nanti kalau misalnya agak malu-malu bisa dikirim di chat aja pakai bahasa Indonesia, nanti saya bisa menyampaikan ke Profesor Malgorzata Korzeniowska. So I think we can start now. So dear Prof. Gosha, the screen is yours.
Hello, everyone. Good morning for us, for me and John sir, and good evening for everyone there in Indonesia. First of all, I would like to thank to John sir and the organizer for inviting me to giving this speech. I will try to share my latest achievements and be a little bit general, so you have the overall knowledge about the mostly biocompounds from meat, but mostly I will focus on the antioxidative components which are naturally present in this kind of material. I will try to share my screen now.
Before that, I'm sorry, Professor.
Some problem with that. Okay.
We highly encourage everyone to open their camera. Thank you.
Okay. Can you properly see the screen?
Yes,
Yes, Prof.
Okay, perfect. So we can start. So as I mentioned, my expertise in the field of science is the antioxidative compounds in the present naturally in meat. So not the one which we can incorporate into the this type of tissue, which is of course also possible. But my main interest is the natural compound, the one which by specific treatment of animals, but mostly by feeding modification, we can also increase or decrease if they are not really beneficial for human consumption. So there is a slight balance in this area between those substances which are beneficial and non-beneficial for us. And then we can also incorporate some other substances, but these modifications are possible, of course, but I'm not really interested in. So today I would like to share with you some aspects of the bioactive compounds present in meat. But of course, the subject is really wild, and the time which was projected for this lecture is not enough for sure. So I will share just the general things. So a little bit of benefits, risks, and challenges of natural meat antioxidants. So as we know perfectly from our basic school and then through our whole education pathway, the oxidative of the general biological function of meat tissue, which is kind of complex, it's not easy to understand, and those pathways which are naturally occurring there are kind of complicated. So we will not go through it, but just to let you know that talking about the antioxidants or any kind of biological compounds present in such complex tissue is really difficult. So we can touch one aspect, but we have to be aware that there are so many different conditions which are influencing the specific component composition, structure, biological activity, and of course the activity in this specific tissue. So just to be aware that one of the most important biological activity of the muscle tissue itself, and also with the connection with fat tissue and connective tissue and blood, this system is so complicated that one of the most important pathway is the oxidation, which occurs naturally throughout the whole life of animal, and even after slaughter, the oxidation processes are going even faster, and in this case, they can make more and more damage to the tissue itself, to our final products. If you want to prepare some design and really prepared products, so the oxidation can cause some damages. And of course, after consumption, these particular reactive components which are formed during the oxidation are especially harmful for us. So just to have an idea how the oxidation processes go through the tissue and they hit the specific substances. Mostly when we think about the oxidation, we think about the lipids. This is obvious because they are the most susceptible to the components which are the most susceptible to oxidation, and they cause the most damage in the animal tissues, but also in human tissues. So we have to be really careful with that. Just try to minimize those reactions of the oxidation of lipids, because they usually cause really big damage in the quality, in the safety of the products, and finally in the consumer body itself, going through total damage of our health. So this is really important. But we have to also, when we like talk about the food components especially, so we have to think about the changing, unfortunately, mostly negative changing, or the safety aspects, but also the sensory aspects, the quality, but mostly sensory, which are connected with the taste, aroma, and color. But talking about the oxidation in the food, so we have to be also aware that not only lipids are going through them, so also protein can go through oxidation. And now when you look at the literature, there are plenty, actually more and more papers coming in this area. So the protein oxidation, it has it can have two different effects on the quality of meat and meat products. So they can be also damaged. So the protein, which are the most nutritional substance in the meat, it is of course damaged, because oxidation caused the turnover of the proteins, disruption, and also hydrolysis in somehow. So all these reactions cause the damages of the proteins. So it lowers the quality of the final product, and it of course can cause some adverse effects on our the quality of the meat, the nutritional aspects of the meat, but also on the consumers. But when we talk about the protein oxidation, and the oxidation is limited in kept in the specific conditions, we can benefit also from this part of the biological or chemical activity, and we can improve somehow the texture and functionality, mostly of isolated proteins. So when we talk, for example, about the surimi production, so then the limited and controlled protein oxidation is actually very welcome, because it benefits the final quality characteristic of the product. And of course, we cannot forget about the vitamins, which are very important in our nutrition. And of course, they also go through oxidation, and then they mostly, in most cases, they lose their activity, they lose their nutritive value, and they can also be a part of damage of the color. So that's why oxidation processes are not really welcome in the food production, in the food chain, and also in the human nutrition. They can cause many different bad and adverse effects on our body, as you can see here. So what we can do? If we can do anything or we can't, of course we can. So one of the most important thing is the antioxidants. So antioxidants, as you see from the name, they act against oxidation. And there are so many different definitions of this word of antioxidants. So in general, and the one I like the most is that antioxidant is an every substance which is actually present in a low concentration in comparison with the oxidized substance, which is able to inhibit significantly, so in a big manner, oxidation of this specific substance. So just in the simple word, it means that if we have, for example, like in the muscle tissue, a kind of big amount of lipids, and we can find tiny amounts of any substance which can influence the oxidation of these lipids, we can call it antioxidant. So we have plenty of classifications of antioxidants, different ones based on different conditions and rules. But mostly we can have primary antioxidants and also secondary antioxidants. I will not go through it because this is like a basic things, and I would like to switch more to the real subject, which is connected with meat. But just to mention what is the way of antioxidant action. So you can see on the screen that they can act by many different ways. One of them is complexing of the metals, and metals are naturally present, especially in the muscle tissue and blood, which are like connected with in animal body. And even though we want to remove as much blood as we can during the post-slaughter operations, some of the minerals from blood are still remaining in the tissue, and they are acting there. Mostly iron. Iron is the one which causes the most of damages. So any substance which can halate, so bind the iron and make it non-available for oxidative reactions, which it's called chelators. And one of the most stunning example of that is ascorbic acid. And of course, you can see that, and you can say that what ascorbic acid has common with the meat. So I will tell you a little bit later. So then the other way is to create the protective layer between the substances which can be oxidized and the substances which are less prone to oxidation. So in this case, the most stunning ones are phospholipids, which are naturally present in meat and actually are present in any animal, like animal tissue. And the phospholipids are really, really important for this antioxidant action. So then the next way is to regeneration of the primary antioxidants. So we have primary antioxidants and secondary ones. And the primary antioxidants are present naturally, but they are used very quickly in the case of when the oxidation occurs. So we are losing them very quickly. So in that case, we have the possibility of regeneration of the rest of the remaining primary antioxidants. I will tell you also a little bit later about this, because we have the typical system of the primal and regenerating antioxidants. Then we have the scavenging oxygen. This is the most popular way of removing the oxygen and the oxides, already oxidized samples, like the structures. Sorry, somebody is keep calling me. So the scavenging of oxygen. So when we have the free oxygen, it can be removed from the system, and in that case, the ascorbic acid is the most important one. And then we have the decomposing process of already formed oxidized product, which can be, of course, radical or non-radical. Then we have the absorbing the cause, the indicators of the oxidation processes. So, for example, absorbing by UV radiation. And the last but not least is the quenching singlet oxygen, which is the most important for beta carotene. And of course, this is not the primary antioxidants of meat, but in the very limited amount, we can also find it in the meat samples. Then we have we know already the way how the antioxidants can work. So now we have to try to find the best characteristic of the most suitable substance which can be used and can be considered to be antioxidants. So first of all, and this is the basic for any kind of substance which we can incorporate into the food, which is going to be consumed no matter by people or by animals. They cannot be harmful. Okay, so this is the most important thing. They cannot be harmful. So they cannot influence negatively any physiological conditions in our or animal body. So they cannot cause any pathological, carcinogenic effects. They cannot interrupt the natural pathways, biochemical pathways, and they cannot interact with other beneficial substances or some even cells in the body. Then they cannot change any of the sensory properties. Mostly when we use the antioxidants, we of course use them in a very small amount, but still if they cause any of the changes in the odor or flavor or color or taste of the product, we cannot accept them. Then, of course, they have to be, according to the definition, very effective in the low concentration. There is no point to use a lot of antioxidative components, trying to incorporate them to the product, because they are not playing their role anymore in that if they are in a high concentration. Mostly they should be fat soluble, but mostly this is not the like the border condition, because we also have the lipid, the water soluble substances, which are really good and active antioxidants. So they actually, the best is that if they are able to act in both conditions, in the fat solutions and in water solutions. And we have the examples of that. For example, phospholipids are the best example. And of course, they have to be active, and they have to be relatively cheap, because you know that economy is one of the most important thing in the world, if not the most, for at least some of the people. So we cannot spend a big amount of our budget when we produce the food on only on antioxidants. So they have to be economically proven. And of course, then the last point, that they cannot be absorbable by the body. So they have to be like either metabolized or just go through the consumer body without any absorption in it. So in meat, we have natural systems of antioxidative protection against oxidative reactions. And one of the most important when we talk about the living animals are antioxidant enzymes. And enzymes, as I mentioned, they are active when there is abundance of nutritional substances, abundance of oxygen, of some minerals. So they work effectively to protect animal body and our body against oxidation. Most of them, like the most important of them, are glutathione peroxidase, catalase, and superoxide dismutase. Of course, there are more, but those three are playing the biggest role in protection. Then we have the big number of proteins and peptides, which are able to act antioxidatively, mostly by their halating properties, so binding the metals or other indicators of, like chemical indicators of oxidation. And of course, we have the low molecular weight substances, which can be hydrophilic or hydrophobic, and they mostly disrupt free radical chain reactions. And these are, you have the whole, the most stunning examples here, like vitamin E, which is totally connected with animal feeding and also animal tissue in the like hydrophobic solutions. And also we have like ubiquinone, which is mostly connected with the water. And then we have also in living animals enzymatic system, which is responsible for reparations of the big, like very big macromolecules like DNA, lipids, and proteins. So we see that actually the muscle tissue is really well protected against oxidation processes. So nature is the biggest, as the most ideal creation in the world. And if we want to like adapt it, we have to be really, really careful, because when you deal with the meat, so like when you take the meat out of the freshly slaughtered animals from the carcass, and then you keep it in the proper conditions without exposure to light, like a very intensive light, high temperature, or there is no contact with the metals. So meat cannot be cannot go through oxidation processes very roughly. But then when we go to the meat processing and we interrupt, we damaged actually the tissue, the muscle tissue. We mix it with the lipid tissues. We mix it with blood. We make the contact with the metals. We expose to light. We add some other substances to make the sausages or other meat products. We damage all these mechanisms, the natural mechanisms. Of course, not all, but the antioxidants enzyme, they are not working anymore. They are not present there anymore. And the enzymatic system for macromolecules is not present there anymore. We still have some proteins, peptides, and the low molecular weight antioxidants, but we have to be really careful because their power is also lost during processing. So just to talk a little bit more about those antioxidative enzymes, they are naturally present in meat, and still have some like they are naturally present in the muscle tissue, in the muscle cells, and there's a very complex system on how they work and how they can be restored, because we are losing them, like they're not renewable. Okay, so we have to be sure that during the animal life, they have to be also supported by mostly by nutrition. Yep. So if the nutrition is not proper, we cannot use the antioxidative enzymes anymore. So, but now I would like to just to mention that we have those regular enzymes like superoxide dismutase, glutathione peroxidase, and catalase, which are three the most important, but also they are connected, there are ones connected with the redoxins action, so mostly for not only for the oxygen, but also for example, the sulfur components. They are very susceptible to oxidation. So that's why this the enzyme of antioxidant actions, they are very, very important in the tissue. But I wouldn't like to talk more about this. If you want, we can come back later to this part. One of the most important, as I can say, antioxidant compounds which are naturally present in meat are tocopherols. Tocopherols and tocotrienols. This is the huge group of antioxidants. And why they are the most important in the animal nutrition and animal products which are derived from animals. So this is because the vitamin E is a really tiny compound. You can see the structure on the screen, and the most important are those rings which are in the structure of tocopherols and tocotrienols, and they are the non-saturated boundings are responsible for the antioxidative actions. So Vitamin E is also one of the most widely present antioxidants in nature. Is present not only in plants. It is naturally present in the meat, and not only the big animals, also small animals, the sea creatures. So this is really abundant material. And why is so important? It's the primary chain breaking antioxidant. So it acts both during the life of animals, and also when the animals are already slaughtered and their meat, the tocopherols are able to remain in the tissue, and they are able to act also in the meat tissue. So even though the meat is already in the chill room, the tocopherol is able to work and act against oxidation. Of course, we have plenty of isomers, so alpha, beta, gamma, delta, tocopherols, and tocotrienols. And of course, the most important one, because of the abundancy, is the alpha tocopherol, because this one is the best known, and also because it's the best known, we claim that it possesses the highest antioxidant action. So and also in especially in big animals like cows and also pigs, even chickens, or poultry in general, the alpha-D tocopherol is naturally present in the feed of the animals, and also is used as a natural antioxidant, like artificially added or additionally input into the animal feed to protect actually the fodder against oxidation. But then it's so fascinating compound that it can be incorporated into the animal tissues. So it can break the barrier between the feeding and the blood and the muscle tissue. So it can be accumulated in the body. So this is also a very important aspect. When it comes to the meat, the tocopherols are able to protect not only lipids themselves, like the typical lipids, but also they protect cholesterol, especially the low density cholesterol. So the tocopherols are able to decrease the total cholesterol level in the blood. So this is important information for those who have problems with cholesterol metabolism. And then when you look at the numbers, and you see that the biggest amount of natural tocopherols and tocotrienols are present in sea creatures, like in shrimps. But when it comes to the meat, especially the meat which are obtained from like the domestic animals, which we can breed, so there is also a quite significant amount, which actually even reaches almost 3 milligrams per 100 grams of animal tissue. What is the most important aspects of the antioxidativity of tocopherols is that tocopherol and tocotrienols are acting antioxidatively by breaking the oxidative reactions, especially in lipids, but also in proteins. They can do it in the fresh meat, they can do it in the store meat, they can do it even in the frozen meat. So this is important when we freeze the remaining part, or like the leftovers or the stock of the meat. We can use really the relatively low amount of tocopherols. It's around 0.02 milligrams per 100 grams, and it's enough. And then the tocopherols are also acting as regenerators of other antioxidative substances. Mostly it is connected with enzymes like glutathione peroxidase, but also sodium peroxidase, and also sodium dismutase, sorry. And the alpha tocopherols, mostly alpha tocopherols, limit also the formation of non-desirable color compounds like methmyoglobin, which improves significantly the red color of meat. Next big group of antioxidants which are naturally present in meat tissue and are carotenoids. And carotenoids, mostly alpha and beta carotenoids, carotenes and xanthophylls, act similarly to tocopherols in the lipid fraction. So there are plenty of them, plenty substance which form this group, and they are responsible mostly for the slightly yellow to even dark red color. When you look at the literature, most of the papers will tell you that the carotenoids are mostly present in plant materials, and this is totally true. But in animal materials, you can find also the carotenoids. Carotenoids, mostly beta carotene, are primer antioxidants which breaks the oxidation reaction chain, remove the singlet oxygen, so it even protects against initiation of the oxidation, and also can regenerate a little bit of vitamin E. So they are like symbiotic. They act symbiotically. And when you look at the contents of carotenoids, you can see that mostly carotenoids are present in the in fish tissue, or even in the sea creatures, the other sea creatures than fishes. But mostly when you look at this rainbow trout or salmon, this amount, like even 25, like you see in the rainbow trout, 25 milligrams per kilogram of carotenoids, it's a huge amount, and it's maybe not comparable with the plant materials, but in some cases, of course, yes. These are mostly connected with feeding, because beta carotene is widely used in the fish feeding. When we have the breeding plants for fish, we like them, especially for salmon or rainbow trout. We really like to have their meat in a nice pink color. So in that case, the breeders are incorporating some of the pigments into it, and pigments are coming from carotenoids. When it comes to big animals, so the concentrations is much lower, so way lower. So you can see like in beef, they are between 0.03 micrograms per gram and up to 0.2 microgram. A little bit more in the total carotene, and like up to 0.3, 0.4, but not more. The one example which is really stunning, it's the pork liver. Pork liver, you can see it has 540 micrograms of beta carotene in each gram of the liver tissue. In the beef liver is much lower. It's around less than 10 micrograms per gram. And of course, in the poultry, the concentration of carotenoids is relatively low, especially in the white type of mussels. So these type of mussels which are less in fat, so they also contain less carotenoids. And the next group of antioxidants which are naturally present, and actually they have a big potential, biopotential, are coenzyme related, coenzyme Q10 related materials. So these are mostly ubiquinones. The most the most recognized one is coenzyme Q10, of course, but so typical ubiquinone, but there are more substances in this family, so they are naturally active in the animal tissue while the animals are alive, but also they are staying active while the meat is already, I would even say, on our table, because ubiquinones are quite stable, so they are not even easily removable from the tissue during some operations. Of course, we have to be aware that the concentration of ubiquinones are not really high in the tissue, but they are there and they act very efficiently. They are a part of the enzymatic system, so mostly connected with the scavenging of radical oxygen substances, but also action. They act in the enzymatic and non-enzymatic system. When it comes to the concentration, we have different concentration in different part of the animals, but the most abundant ones are the chicken heart. So actually in the, because I have some small experience in Southeastern Asian countries, and I know that you use all the parts of chickens, for example, and poultry in general, but chickens, and you consume the heart. So you see that the chicken heart is really abundant in the coenzyme Q10, mostly around more than 10 milligrams per 100 grams of chicken. And the chicken meat in comparison has less than 10 times lower concentration of the this type of antioxidants. Where we can find the coenzyme Q, active coenzyme Q in other type of meats, fishes. So again, because you're famous from the big consumption of the fishes, so you do a really good job, because like in sardine and mackerel, we have more than 5, 6, even up to 10 milligrams per gram per 100 gram of meat. The other group of the very active antioxidants, it's conjugated linolenic acid, which is present not in all animal tissues, but is strictly connected with animals. We cannot find it in any other materials, like plant materials are not present. Of course, the conjugated linolenic acid is present in the ruminants, and this is typically for this type of animals, so sheep and cows, goat, only this type of animals. And the conjugated linolenic acids is a kind of the isomeric forms of linoleic acid, which is actually present everywhere. So in different concentration, of course. But mostly the biggest amount of these particular antioxidants are present in the red muscle. So you can see around 10 to even 50 milligrams per 100 grams in the raw meat. But of course, we don't consume the raw meat at all, even though in the very low concentration. So we usually like we do the thermal treatment, any kind of thermal treatment to the meat to make it more edible for us, more attractive for us. So especially in the red meat, the concentration of conjugated linolenic acids are increasing up to 30 to even 100 milligrams per 100 of grams. The other group are the phospholipids, which are naturally present in the cells, also in the muscle cells. So the most important ones are, of course, lecithin. They are the most famous, the best recognized. But the phospholipids are not single lecithin. There are much more, and you have this complicated names like singomyelin and lisophosphatidylcholine and inositol and their derivatives. So they act, they are the very unique one because they are able to act in B layers, like they have the activity, they express their activity either in hydrophilic and hydrophobic layers and their conditions. So which is really important when we have such complicated system like meat. And especially with the phospholipids, we have to be really careful because when they are present in the low concentration, they act typically typically antioxidatively. So they protect the either lipids or protein against oxidation. But if we want to use the additional portion of phospholipids and incorporate the phospholipids into the system of our meat product, we have to be really careful because when we cross the line, cross the border, we can do more damage than positive aspects, positive effects, because phospholipids can easily transform themselves for typical pro-oxidants, so they can even promote and speed up the oxidation processes. And vitamin C, finally, we reach the vitamin C, which is typically present in the plant materials, which is really abundant in the plant materials, which is one of the most important substances in the human nutrition, because if we have some limitations and lacking the vitamin C, our organism is already showing us that something is wrong, mostly in most cases. So in general, when we think about animal materials, so meat, fat, or even milk and eggs, people will say that just the regular people which are not dealing with science, they will say that no, the vitamin C is not totally present in this type of materials and products, which is of course not true. And we as students of the food technology, food science, or any kind of related science or related fields, or and even more, we scientists, we have to share the knowledge that the ascorbic acid is naturally present also in the animal tissue. Of course, the concentration is really low, but it's not the truth that the vitamin C is not present there in any case. So we have to show people that the vitamin C is present in the animal tissue, and it's one of the most effective antioxidants. Of course, because of the concentration, it has a limited role in the natural meat system. But then when it comes to the product, and we additionally apply the vitamin C, mostly by sodium ascorbates, into the meat systems in the products, so the concentration is higher, and this antioxidative actions is really, really, really important. So how the vitamin C actually acts, I think that you know perfectly. It slows down the mostly lipid oxidation. In protein oxidation, it is questionable. It depends on the conditions. But in lipid oxidation, in most cases, the vitamin C is able to slow down by mostly direct scavenge of superoxide, hydroxyl or lipid hydroperoxide radicals. But also vitamin C can halate metals in a like very limited extent, but still it is possible. And of course, the vitamin C acts synergistically with vitamin E, and this vitamin C is able to recover vitamin E, so the tocopherols. So this is important, because if there is no vitamin C at all in the system, the vitamin E, so tocopherols will be lost very quickly, removed from the whole system, and we will lose it, and the material will be really prone to oxidation. So it is important to keep the vitamin C level in the living cells on the specific level, the required level, and of course, after when we go to the processing part, it is important to add additional portion of the vitamin C into it. When you look at the concentration, the natural concentration in blood, for example, it's around 40 micrograms per 1 liter of blood. When it comes to the meat, is around 9 to 16 in a dark type of meat, like red meat. And in light meat, it's like a little bit higher. It's even like 24 milligrams per kilogram of the mackerel light muscle. Then we have glutathiones. This is also the group of substances. Mostly acts by the remaining and stimulating the enzyme system, the enzymatic system. So we have this natural working mechanisms for protection of antioxidants and the tissue itself from oxidation, but also the glutathione can act synergistically with both vitamins like C and E, so ascorbates and tocopherols, and of course they can act, like they can play some roles in the synthesis and reparation of DNA, and also protein synthesis, amino acid transport. So it has plenty of different aspect in the living animals. But then when it comes to the meat tissue, the glutathione, which has this specific composition of 3 main amino acids like cysteine, glutamic acid, and glycine, the glutathione works mostly on breaking the chain of the oxidation reactions. And when you look at the numbers down there, the glutathione concentration in meat, especially in fresh meat, is very different, like varied from around 1 milligram per 100 grams up to even 26, 30 in beef. So like when you look at this, so chicken and beef are very nice and good sources of glutathione. Of course, when we process the meat, the glutathione stays there in major concentration, so it's not lost very easily. So in the thermally treated meat, the concentration is higher, but mostly because the dry matter of the substance of the product is higher. Then we have the really small substance which is called alpha lipoic acid. Is not very common in the other materials than the animal world. So it's like vitamin-like antioxidant. So the structure is similar to vitamins and is mostly complexes the minerals, and also it's able to break the chain of oxidation reactions. So the concentration is not very stunning. It's around 1 to 3 micrograms per gram of dry weight of the muscle materials. One of the most abundant materials of the when it comes to lipoic acid is their offals. So offals are really nice to consume, either by just the thermal treatment, or when we incorporate them into the like meat products. So then I know that not all over the world the offals are consumed, but like here in our country, as I know in Indonesia also you consume the offals. So this is really really beneficial material, and it bears actually the high nutritive value. And then we have the whole bunch of amine compounds like amino acids, peptides, and proteins. And it in that case, the amino acids are naturally present in the meat. They actually build the muscle tissue, but also they can act antioxidatively, mostly by regeneration of primary antioxidants. And actually all of the amino acids has some antioxidative power, but the biggest significance are put into those ones which are mentioned. So tyrosine, histidine, methionine, lysine, arginine, phenylalanine, and tryptophan. One of the most interesting one when it comes to the amine compounds, it's taurine. Taurine is so-called fake amino acids because it's not really in this group of 20 basic amino acids, but it belongs to this group. And actually it's the most the highest concentration is found in the meat. And when it comes to the taurine, which is derived from methionine and cysteine metabolism, it really plays the significant role in the antioxidative protection of the muscle. And meat, when we consume, for example, in sheep, especially young sheep or lambs, even 110 milligrams per 100 each 100 gram of muscle tissue. So taurine is also very important when it comes to actually isolation, and also the incorporation of pure taurine into the other products, because like in the European Union, when we produce the food for babies, for young people, but especially newborn babies, infants. So the use of pure amino acids is forbidden, and only this one, only taurine is like available for, and it has the permission to be used in the infant food. So we can even isolate the taurine from meat and try to enrich the baby food into the taurine. Actually, when we talk about the amine compounds, one of the most important are antioxidative peptides. Mostly because they act as free radical scavengers, and the most important ones are carnosine-related compounds, which are, of course, naturally present in meat and only in meat. They are not present anymore in any other materials. So we have the carnosine and anserin, which are the two most abundant ones. They are found mostly in the skeletal muscles, but also in offals, but also in like brain. And why they are such important? Because there are plenty of papers on that, and the carnosine and anserin and their components they derived, they play a significant role in the protection of Alzheimer's diseases. So we have to be sure that our natural level of carnosine and anserin, or generally carnosine-related substances, we have to keep it on the good level. So why? Because carnosine-related substances are or compounds. They buffer the muscles. They lower the toxicity by halating the metal ions. They play antioxidative roles, mostly by scavenge, as I mentioned, scavenge the oxygen species, and they also scavenge the low molecular mass aldehydes. So they act against anti-glycemic. And of course, they are the precursors of other substances which are important in the protection of our health. So in that case, we can see the concentration of the carnosine and anserin in different substances, in different products. So the most abundant in case of carnosine are horse legs. Okay, so I know that the horses are not the horse meat are not consumed in all the countries, but when it comes to the other more popular sources of meat, so beef and actually pork are also abundant in muscles are also abundant in carnosine. When it comes to anserin, the white type of muscles are more important. So when you look at the chicken breast, for example, it's even 32 milligrams per 100 grams of anserin, and turkey breast, and it's even higher. So the thing is that we can incorporate those carnosine and anserin into our nutrition by, for example, not even consuming the whole meat, because the carnosine-related substances are dissolved in water, are soluble in water, so we can, actually the biggest amount of those substances are in the solutions which are obtained during the thermal treatment. So when you prepare the chicken broth, for example, or turkey broth, or the beef broth, you can it's really beneficial to eat the broth, not the meat itself, just a broth. And when you have the older people in your family, so just encourage them to eat a lot of broth, actually the meat broth, no matter the type of meat, but broth. So the solution, the extract of water-soluble compounds from the meat. This is the best for their memory. So if they want to save the memory, this is the best. And we have the carnitine, which is present in other materials also than meat. But the carnitine in the low concentration is mostly connected with the energy and the vital power of the meat. So mostly if we want to use this type of antioxidants in our nutrition, we can consume the sheep, so meat, so like lamb and sheep. So even up to 210 milligrams per 100 grams. In beef, around 60 milligrams of 100 grams. And then creatine, which are connected with the carnitine, they both play very important role in the energy metabolism, but also they express small but still express the antioxidative power. And the biggest amount of creatine is usually found in the red meat, mostly in fishes or pork, beef. In poultry, not the highest amount. And one of the last antioxidant which is present naturally in meat, it's selenium. And selenium is tremendously important because it regulates the enzymatic system of glutathione peroxidase. It's one of the initiator and stimulator of the creation and synthesis of glutathione itself, and also it's a part of like the natural metabolism. And selenium as a mineral is also a very strong antioxidant. But there is one the very stunning thing about the selenium is that selenium itself is a toxic material. So we have to be really careful when we want to incorporate additional portions of selenium to the animal nutrition, because when we exceed the level of selenium, we can kill the animals. Or we can really damage their metabolism, so they will not be able to grow and function properly. But in the other case, the selenium is the crucial point in the animal nutrition, and without the proper level of selenium, the animals will not grow at all. So we have to be really careful about this. So when it comes to selenium, the animal parts of our nutrition, so the sea creatures like oysters are very abundant in selenium, not so the tuna, but even in the pork and beef and even chicken, mostly the red meat are kind of high in the selenium content. In the polyphenolics, this is the last part. So polyphenolics are very famous and popular, and in the last years, most of the people in the scientific area of nutrition were very fascinated in polyphenolic substances. So they try to incorporate polyphenols into the animal nutrition. And of course, the biggest amount of polyphenolics, the very wide range of polyphenolic substances are present in the plant materials, mostly in red and dark color fruits, but also green leafy plants. So we consume it every day, and I hope in a big amount, because these are the really good sources of polyphenolic substances, and the polyphenols are amazing, tremendous antioxidants. They act on almost all possible way to break the oxidative reactions, to stop it, to protect it. So we need to eat more and more polyphenolics, but from the natural sources. And when it comes to the meat doesn't have polyphenolics at all. And it's not possible to incorporate the big structure of polyphenolic substances into the animal tissues, because of course this is really beneficial to use the polyphenolic substances into the animal nutrition, because the animals can benefit from it, and they can improve their health tremendously, their welfare. So we should incorporate the polyphenolic substances or materials which are rich in polyphenolics into the animal nutrition, but we cannot expect that the polyphenolic substances will be transformed into the animal tissues. This is impossible. This is physiologically impossible. So in that case, the best combination is to eat meat with some plant materials. So just to summarize, how we can improve the natural antioxidants, the concentration in the animal tissues. We can do it by enriching the animal diet. We can also try to, or this is actually the biggest and the most open possibility. We can incorporate the antioxidants into the food while processing, and we can also fortify the food. And we did some of the experiments on that. I will just give you 3 examples. So one was incorporation of carnosine, histidine, and beta-alanine into the chicken and turkey nutrition, and we obtained even 30 times more carnosine-related compounds in the chicken broth than in the regular control animals. In the calf, just incorporation of the selen into the calf or beef nutrition affects the stability, the lipid stability during storage. So we can have one of the example. And of course, during the TBRs is one of the typical indicator of the antioxidant of the oxidative spoilage during storage. So we can see that it was the limited storage. When we did our experiment on selenium, so we incorporate selenium into the chicken nutrition, and we got different results, but the most stunning one was that selenium, especially in the organic form, it increased the carnosine and anserine level in the chicken, mostly chicken breast, but also chicken legs, but it's a little bit lower. And also when we wanted to increase the taurine level in the chicken nutrition, nutrition, we fed chickens with the additional portion of selenium, so the taurine level was increased significantly in the red muscle, so in legs, but no effect was observed in the chicken breast. And also with the ubiquinone, some effects we obtained with the incorporation of selenium into the chicken nutrition. So at the end, I leave you with the questions, because now we have the different trends and with the nutrition, the human nutrition. Many people say that meat is not healthy. Meat can damage you. You cannot eat meat. It's better to switch to the vegetarian diet. But the question is meat to eat or not to eat. So you can think about some aspects, as I already mentioned, and decide by yourself if it's beneficial to eat meat or not. Thank you so much, and I think I exceed the time, but okay.
Thank you very much, Prof. Gosha. And if any one of you have a question, you can speak or you can write in the chat. Kalau ada pertanyaan bisa ditulis. Mungkin malu pakai bahasa Inggris nggak apa-apa, dari tulis pakai bahasa Indonesia juga nggak apa-apa.
Okay, I want to ask you.
Okay.
Yeah. Yeah. Can I direct set my question?
Yeah, sure. I think.
Yeah, you can send it by chat.
Okay. Yeah. As we know, there are some commercially such as selenium, carnitine in the market. Then I want to ask you, what is the difference, or maybe what is better we consume, the commercially that we can buy, or we eat just meat as a source of the these important things for us? Thank you.
You mean that on the market there are plenty of like
Yes. Plenty of selenium, carnitine, and so many. Even coenzyme Q, ubiquinone. Yeah, we can find it in the market.
Yes.
What is the difference, and what is better we consume?
That's a good question. You know, because these supplements, which you mentioned, they're of course very concentrated. So if you have a very fast effects on your body, of course, I wouldn't say that it's better, but it's faster. The main difference in that case between the supplements and the natural materials like meat is that the supplements are mostly produced synthetically or on the microbiological way. So the concentration is higher, they act faster, but the availability is lower. So we have to consume more to have the same effect like when we consume the natural sources. So in that case, it's better to consume meat than with a longer period, of course, and not very big amount, which is not healthy, as we know. But it's better to consume, like to take the natural substances from the natural sources.
Okay, thank you. I'm afraid also preservatives that they use. Yeah, because
this is the thing that in these supplements, they usually, when you look at the composition, you see that the active substance is actually not very high in concentration, but they have to have the base, like the carrier in which the active substance is incorporated to take it, because the concentration of active substance is really, really small. So, and we will not be able to see such a small concentration. So they have to put it to the carrier, and then we have to consume it. So, and then of course the preservatives they use because they have to be stable.
Yes.
And they have to protect the active compounds which is inside. And in many cases now, not even now, but it's the regular rules that they have to be stable and protect the active substance when it goes through our digestive tract. because the active substance should be released in the intestine. So it has to be able to survive the very rough and strong conditions in the stomach. So
Yes.
that's why the supplements are, you know, okay for the short time when we want to have the effect, but consuming them every day for a long period is not really recommended.
Yeah. And the taste, I think. The taste is better. Yeah, when we consume the nature one. Okay, thank you.
Yep, thank you.
Thank you very much. Is there any more questions?
Yes, may I have a question for you?
Yes, sure.
Can you, audible? Okay. It is, thank you so much for your presentation, and this is very exciting. Actually, nowadays, due to the pandemic COVID, everybody right now, especially in our hometown, taking the vitamin and supplement in a very high dose. And, and sometimes even to find the vitamin C or other supplement is very hard right now. So based on your presentations that now we believe that natural sources of the vitamin is more efficient and then more healthy, then we take the synthetic supplements. But I would like to ask you, what do you think if for the long term we eat more protein than carbo? Because nowadays also is very popular like a nutrigenomic dietary, and then also the keto dietary. What do you think about this? Thank you so much for your time.
Yeah, I'm not a nutritionist, but as my knowledge allows me to say that the best nutritional recommendations is to eat everything, but in a small amount, like in a really well balanced amount. So it's not from our body point of view, from our nutritional point of view, it's not really recommended to stick to only one group of nutrients. So because if we need everything, so it's like some people say, and some even doctors says and recommended that people shouldn't eat food which increase the cholesterol. So in that case, this is not really a proper recommendation, because in general, the human population has no problem with cholesterol. So, and the other thing is that biologically, even though we eat the food which is rich in lipids and cholesterol, they are not connected with our cholesterol level in the blood. Of course, when we are not having any problem with the metabolism. So in that case, we need cholesterol, because the cholesterol is natural compounds in the bilayer system of the cells. And this is, you know, it's impossible for us to live without it. So when we skip totally the lipid compounds from our diet and we stick simply to proteins, the proteins are very heavy for our nutrition, for our digestive tract. So it's okay, it could be done for a very short of time, but when it's for a longer period of time, our body will change and will actually ask and require some lipid compounds or carbohydrates. So it's totally not recommended to use only one group of nutrients in the long-term nutrition. Yeah, and of course, coming back to the first part of the question is that it is recommended by some doctors, especially in the COVID period, to consume or to actually it's not consumed, but it's not consumption, but to take more supplements, like vitamin supplements. Of course, when we take the supplements, as I said before, they are concentrated. So we have the high concentration of the active substance in the short time. So, but again, we have to be remember, I think I mentioned it only once, but I should more. So even with the tocopherol, tocopherol is really, really good, or even vitamin C, they are very good vitamins, and we can eat many of them, like now to a certain level, and they protect our body, they act properly, they improve everything, our daily life. But when we exceed the amount which is proper for our body, they start acting against us. They act actually with the pro-oxidatively, and they change the metabolism, they change some biochemical pathways. So this is not really recommended, again, for a long period of time to consume a very high doses of vitamins. Especially we have to be careful with the lipid-soluble vitamins, because of course, when there is a deficit of them, we suffer. But when we have exceeded amount, we suffer again. So this is really, it has to be really well balanced. I think it was in the 90s, in the last century, the vitamin E and generally tocopherols were really popular, and there was the trend to promote the high consumption of vitamin E. So people were attacking the the supplements that they took a huge amount of vitamin E, because there was the saying and some of the research done that the vitamin E is able to protect us against cancer, any kind of cancer, which is actually totally not true. Because of course, when we have the well-balanced, the proper level of vitamin E in our body, it's okay. Yeah, so this is protective role. But when we start taking too much of vitamin E, it goes totally adverse way. So we have to be really, really careful.
Okay. Thank you, ma'am.
Thank you very much. Do we still have time? Pak Adrian, Bu Elisa?
Yes, of course. Until
So
16.
okay. So if still there are some questions, please ask.
If you have more questions, you can send it also to John Sir or to my email directly, and I can answer them.
Yeah.
Maybe I want to ask to Professor Malgorzata, Mr. John, sir.
Yeah, go ahead.
Okay, thank you. Thank you for the excellent speech for the bioactive compound in meat and meat products. And my question is, in vivo study, which you shown to us before using experimental animals of various types, which animal is the best used to get the accurate result? Because you saw about many animals such as horse, ox, and I don't know what is ox, pigs, and other animals. Because if we want to use an like an in vivo experimental here, we just use like a mouse, white mouse. I mean. So what is the protocols to use them? I mean, how many animals? Example, you use horse. How many horse you use to get the best result? Thank you.
Thank you for this question. I presented some of the results which were done on horses and oxes. Oxes are the bulls. Okay, so they generally beef, the cows and bulls. But these are not my results. So I took them from the literature. And of course, when you make I did my results on chickens and on turkeys. In general, the research layout depends what you want to obtain. When you want to study the availability of some substances from the animal tissues, or how the biochemical transformations are going there, or some biological aspects, of course, it's the best to use mouse or rats, because they are actually the closest to us. Just it looks strange, but it is biologically. But when you want to perform the nutritional study from the animal point of view or human point of view, it's better to use the animals which we consume. And until now, the consumption of mouse and rats are not very popular, especially like maybe in Africa, but they are not, you know, they are not the popular animals like for this type of studies. So actually, chickens are the very best animals for the research. Maybe I wouldn't say the best, but they are really, really good in some cases. The first of all, they grow very fast, so you can finish your experiments even in 4 weeks. You can try different modifications in the feeding system. So the chicken responded actually very quickly. So this is the good material for the experiments if you want to have the results. Usually when we do the experiments on chickens, the smallest number of animals which has to be taken to the from the statistical point of view is like 30-40 per experimental group. And of course you have to repeat it just to be sure if everything was okay. And when it comes to big animals, of course we can use it, but the number of animals are similar. But so the lowest number is like 10 to 15 in the big animals, like cows. In pigs, around 20. But these are just the general numbers. It depends, you know, the animal the experiments on animal, even the nutritional ones, are very expensive, and they require the ethical commission approval. So it's not really easy to get. But again, in this case, the chickens are actually the best because we consider them to be like, you know, but small animals. So they are not that demanding as the big animals. So that's why, and actually we are working on poultry. So the chicken and turkeys are the good materials. Turkeys are much more susceptible to changes in the environmental conditions. They are really delicate animals, so we have to be careful with them. So again, chickens are more stable, are more resistant, are fast growing, and they give us the very good results.
Okay, thank you for your question. Any more questions? If not, as Prof. Kosya said, you can send me or to her direct an email, and then she will answer. Yeah.
Okay, so if there is no more question, I would like to thank you all again for your participation in this lecture. I hope you enjoy it and you get some information, or I spark you with some new ideas to look for some new areas of research. And if you have any question, feel free to send it to Joser, and he will transfer the questions to me, and we will transfer them back. So thank you so much. And as I have the other meetings, I will say goodbye. Have a good evening, and hopefully to see you one day in the reality.
Thank you, Dr. Malgorzata.
Thank you.
Thank you very much, Dr. Malgorzata.
Okay, so I will give it back to the Prof. Elisa.
No, John, sir.
Ya,
No.
berarti balik ke MC yang tadi ya, Bu?
Ya, ke MC.
Oke. Saya kembalikan ke MC. Maaf.
Baik, thank you very much for our speaker. That's a very attractive and a good information for us. To close this event, I welcome the head of Food and Science Technology Government Faculty of Agriculture, University of Sumatera Utara, Professor Dr. Insinyur Elisa Julianti, STP MSI. For Mrs. Elisa Julianti, the time is yours.
Okay. Can you hear me?
Yes, clearly, Prof.
Okay. Thank you very much for Dr. I forgot your name. Thank you very much for Associate Professor Dr. Malgorzata Korzeniowska for the nice general lecture today. We have many information and knowledge from your presentation, and thank you very much. See you next time in the actual meeting maybe. And thank you also for John Sir for your help to us to this general lecture today. Okay, maybe we can close this general lecture today, and once again, thank you very much for all the participants today. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.